And for the nice introduction, and thank you actually to the organizer for bringing both, both champion and POC scientists and families together for this amazing event. We learned a lot actually, so it's a nice brainstorm for us as scientists. I would like just to introduce a little bit my lab and what we do before handing over the microphone to Daly, our postdoc, who's going to present actually the data on champion that we have in our lab. So our lab focuses on two primary areas: a basic neuroscience approach aimed at covering the role of champion in brain development, in specific neuronal development, and like a recent actually translational neuroscience approach dedicated to developing and testing therapies for champion syndrome. For the basic neuroscience approach, what we use is what we do is actually using patients-derived in vitro models, specifically induced pluripotent stem cells. We reprogram those in neuroprogenitors, and then we differentiate those in neurons. We then use electrophysiological and neuronal connectivity approaches, such as whole cell patch clamp, and soon also multi-electrode arrays, to actually identify the electrophysiological properties of champion neurons, to identify the differences between the champion neurons and actually neurotypical neurons derived from neurotypical donors, and of course also to understand the network between these neurons and how champion plays a role in that. And next, also we use genomics, so in this case a single-cell omics, to identify the transcriptional programs that are affected by champion, and how these correlate with the electrophysiological properties that we identify with the whole patch clamp. For the translational medicine, actually, we have two exciting collaborations. While with Dr. Joseph Baxbaum at Mount Sinai, he developed an ASO therapy. For those who don't know what is an ASO, these are like small antisense oligonucleotide that can bind the RNA and pulling down actually the target RNA. In our case, what we do is actually targeting one specific mutation, and we are going to apply this kind of therapy soon. And the other actually exciting collaboration is internal at MUSC with Dr. Chris Cowan at MUSC. We are developing actually an RNA therapy, but he will talk about this like in the next presentation. So, like just a quick update before handing over the microphone to Daly. Where over the past year, I started to work with the champion basically 1 year ago. We started to collect a lot of neurotypical iPSC lines, but also like patients-derived iPSC lines. We have multiple lines carrying different mutations. Daily work will focus on these particular two, one nonsense mutation and one line carrying a frame shift mutation. The other exciting news, and one recent update, champion went to Neuroscience 2024. This is actually the biggest neuroscience convention in the world, more or less 25 attendees this year. And Daly presented this like our project on champion, and she was basically overwhelmed by the crowd, so pretty cool. And as far as I know, like this is the first time champion goes neuroscience, so it's a nice win for us and also for the community. So. And the other, like recent nice update is actually we got our R1, the first R1 funded for champion, for the champion in neurodevelopment. So what we are going to do, this grant will fuel actually like our research on champion and the human brain development and function. So in this case, I would like to thank, of course, Daly and Christina here with us. They are like both did like an amazing job of collecting the data and provide preliminary data to convince NIH to fund actually our project. But the other thank you goes to the family. Sorry for the tone. For the family, because without you guys, we couldn't achieve this. So we thank you for the blood samples. Thank you for the iPSC lines that we've been able to collect, because, as I said, without you, we couldn't do this. So thank you very much. So now I can hand over the microphone to Daly. She's going to talk about the role of champion in neurons. We have like a lot of electrophysiological data from these two lines, so she's going to be explaining everything. So thank you very much.